Well, hello and welcome to your PowerPoint for Bio 112, Chapter 25. So let's go ahead and get that PowerPoint going and started. Your Chapter 25. It is the origin and history of life on Earth. So in our previous chapters, we discussed two concepts, microevolution and macroevolution. If you recall from previous chapter, microevolution is evolution at the level of the alleles. And so macroevolution, that's going to be a broader pattern of evolution, and it's above that species level. And so in that fossil record, when we think about the fossils that we find, those are not really going to be able to point to the alleles and the genetics. They're really going to be showing that morphology that can basically paint a picture of macroevolution at a higher level, above the species level. And definitely a big level that we find ourselves is around the phyla level or the phylum level. And so your fossil record is going to show those macroevolutionary changes over large scales of time. And so examples, we get to see, for example, the emergence of the terrestrial vertebrates. The thing about land is a better conducive environment for those vertebrates to be able to be preserved versus in the water. Now, with that, you also need special conditions. You need anaerobic environment, most probably. That's the best environment for it. And then also to be covered in with lots of heat and pressure over a long amount of time. So with that, in the ocean, not the best unless it's falling to the sea floor as it dies, gets covered with lots of the ocean floor, and then puts it in an anaerobic environment. And that is that more perfect environment. So you were able to, in the fossil records, see a lot more terrestrial vertebrates. And so you've seen those vertebrates arise. You also get to see the impact of mass extinctions. And so throughout that fossil record, you get to see large changes and then a lot of organisms becoming extinct and basically disappearing from the fossil record after a specific time. Next would be, and a good example would be an origin of key adaptations. So as you look at the fossil record and go through time, you can actually see changes such as the flight wings evolving. Now, it is not a step-by-step process. It is a step-by-step process in reality, but in the fossil record, you're going to see big jumps where it wasn't present and then all of a sudden it is present. But you can see that consistently all throughout the earth in different strata. It is very consistent. So conditions on Earth, early Earth, made it possible for the origin of life. And so your chemical and your physical properties of early Earth, that made it possible to produce what was considered at the time very simple cells. They weren't quite cells yet, okay? But it went through a sequence of stages. And so the first stage would be abiotic. So when we say abiotic, we are talking non-living. So abiotic synthesis of small organic molecules. And so that is the making of carbon, the making of hydrogens. I don't mean the making of them, the putting together of those small molecules. So abiotic. So specifically, we've talked about this before, the bonding of CH2O. This abiotic synthesis of CH2O, that right there is a precursor to a glucose molecule. Number two, the joining of these molecules into macromolecules. Well, there's the precursor. Let's times that by 6, and you have yourself a C6, H12, and an O6. This right here is a glucose macromolecule. Now I'm going to actually hand over permissions to my iPad so I can go ahead and do some better drawing on this. All righty, perfect. So the next level of making cells would be the packaging of those molecules into what we're going to call pre-cells or protocells. So you may have heard them called pre-cells. Really, we're going to call them protocells. And that eventually led to the origin of self-replicating molecules. Self-replicating molecules, your DNA and your RNA molecules. Those are going to be your replicating molecules. Okay, so let's go ahead and start with the beginning of Earth. How about that? So these numbers, by the way, some of these numbers, I'm gonna be like, hey, you really should know these numbers. This is one of those numbers that you should know. Earth formed about 4.6 billion years ago. So that is a very significant number that I say is worthy. And so basically, bombardment of Earth by rocks from space and your ice vaporizing into vapor water, that prevented the seas from forming until about 4 billion years ago. And so your Earth's early atmosphere did not have very much oxygen at all. It likely contained water vapor and chemicals. So those chemicals, for example, your carbon dioxide, your CO2, lots of CO2. So your water vapor is going to be your H2O. You're going to have your hydrogen gas, your ammonia, NH3, and your methane, CH4. So your atmosphere on early Earth was very, very toxic. It was nothing conducive to the life that we need. So I should have a nice, pretty picture. I'm going to go ahead and clear this and find us our beautiful internet picture. Let's get you a new share. Oops, I'm sorry about the doggies. So it was already there because I just looked it up. Prehistoric volcano. I'm going to find that perfect image. So this is funny. This is what I'm talking about is before organisms came about. So as you're looking at this and you see my dinosaurs looking like they're going to drink out of a hot sea, yeah, that is definitely not the picture I wanted to depict, okay? Because the timeline does not add up. So let's go ahead and open up this image. I like this image right here. So what we're looking at is we are looking at the Earth prior to life being made on Earth. And so as I showed you in the last, you had lots of toxic gas in the atmosphere. You had your CO2 gas, you had your hydrogen gas in the atmosphere. You had your ammonia, which is going to be your NH3. You also had some methane, CH4, and you also had water vapor. And then in the prehistoric seas, you have water vapor. Okay, so it is hypothesized through the Stanley Urea experiment. They actually went through and they showed how organic molecules could be synthesized from non-living, so from abiotic. So how could that be possible? Let's go ahead and throw some lightning in here. lightning. And that lightning, lots of energy. It's going to break these bonds. Okay, lots of energy lightning. What's it going to do? It's going to break the bonds of the carbon and the oxygen. Going to break the bonds of the carbon and the H4. And the same thing with the water and so forth. And so by breaking those bonds, then when the earth began to cool, those atoms found new bonding partners. So let's go back to our PowerPoint. So those atoms found new bonding partners. Your Stanley Miller did experiment with Urey, Harold Urey, and they conducted lab experiments to demonstrate that abiotic synthesis of your organic molecules, they actually were able to get together and make the CH2O. So let's just go ahead and find his, they don't have it. That's okay, I'll draw it. I'm pretty good. Let's go ahead and make myself a whiteboard. I will draw out Stanley and Urey's experiment for you. So what they did is they had themselves nice little chemistry equipment. Looks like this. Okay. And then from here, give me just a moment with my iPad. Looks like this. Ooh, I should have left an opening. You know, I could go to the internet, but why would I go to the internet when I really love to draw? And so forth. And they have it dripping in. Looks just like that. Okay, so they put their experiment together where they had water vapor in here. I'm sorry, liquid water, H2O. And then from there, they heated it just to simulate that Earth in prehistoric times, lots of heated water. And then from there, you end up with water vapor. And that water vapor traveled like water vapor does. Let's go ahead and throw some lightning on this. So what they did is they put electrodes to simulate lightning. And then inside this top part of their apparatus, they had some CH4, they had some NH3. Remember, your water, H2O, came in. They also had themselves some carbon dioxide and H2. I think that's good right now. So they put the electrodes to go ahead and break. And then from there, let's go ahead and draw a little condenser. So this is a condenser that was put around the apparatus. If you've ever had chemistry, you know what a condenser does. It will input cool water, and that cool water will essentially surround this area right here. And so it will allow those gases to cool. So insert cool water, cooling. There you go. So then from there, you ended up with the liquid going from vapor into liquid. Let's go ahead and draw this pretty like this. There we go. But when they tested the new liquid, what they found in that new liquid were these combinations. They found CH2O. So CH2O, they are abiotic molecules, meaning they are not found in living organisms yet. This is before organisms were present on Earth. This is before bacteria or anything else is present on Earth. So look at that. You got your CH2O. Let's times that by 6. You get 6 of them together, and you got yourself a C6H12O6. And what that is a glucose molecule. And the glucose molecule is one of the very first molecules to come about. And so that is what Stanley and Urey did. They actually took these gases and simulated the prehistoric Earth to show that living molecules could be made from non-living molecules. Okay, let's go ahead and head back to that PowerPoint. So this is everything I've already said. I'm just going to go ahead and scoot through. And so moving on to hydrothermal vents. So your organic compounds may have been produced in those deep hydrothermal vents. Those are areas on the seafloor where hot water and minerals gush from the interior of the Earth's ocean floor. So that they can even test that now and get information now because these are present still. So going into that abiotic synthesis of macromolecules. And so abiotic, remember that means non-living, not living, is that biotic part. So with that, your RNA molecules have been produced spontaneously from simple molecules. So RNA is going to be the molecule that is going to quote unquote carry the DNA's instructions, and then it is going to be used to synthesize proteins from the instructions of the DNA. Well, before DNA was around, RNA was around first. And so your RNA molecules, they thought spontaneously were produced in the same similar fashions that Stanley and Urey have suggested for your glucose molecules. So some organic molecules, they polymerized. What does that mean to polymerize? If you remember from your chapter 5 of your biology 111, to make a polymer is to take smaller molecules and to put them together to have smaller molecules and put them together to make larger molecules. And so they polymerized in concentrations of hot sand, clay, and rock. So your protocells began with the replication and metabolism that are key properties of life. And so it is these protocells that first started making having these systems of replication taken place. And so they may have formed from fluid-filled vesicles. So the thing about a vesicle, and so a vesicle is very simple. If I had a bucket full of phospholipids, I can make myself a vesicle. And so if you think about it here, what is a phospholipid? A phospholipid has a hydrophilic head and hydrophobic tails, if you recall from your Bio 111. And so your phospholipids are going to, if you look in the hydrophilic head, hydrophobic tails, the hydrophilic head has choline, has glycerol. Okay, so it has molecular components of carbon and hydrogen and such. And your hydrophobic tails are going to be your carbon, 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 surrounded by hydrogens. So they're very simple. It's just a matter of bringing everything together. And so with that, if I were to throw a bucket of phospholipids into some water, what's going to happen is you're going to end up with this happening and so forth. Oh, let's go ahead and get some facing the other way. They are just going to arrange themselves into I could be drawing all day because the heads are going to face outwards while the tails face inwards. Here's my little kindergarten picture of phospholipids and how you throw them in water and they will just automatically arrange themselves with the heads facing towards the outside of the cell and the tails to the inside. And so what do we have in the middle here? We have ourselves a loon. Let's go ahead and get some color on it. We have ourselves a loon right here in the middle. So essentially what I have just drawn you is a vesicle. There is one layer of the membrane, and here's the second layer of the membrane. So I just drew it a little bit more with the phospholipids. I'm going to pause this real quick for the moment. If I can get this recording to pause, there we go. Oops, can't do it, I gotta pause it on here. Maybe I don't need to pause it, that might have fixed my problem. All right, let's go ahead and share screen again. Sorry from that. I'm working with iPad and my computer at the same time. So I can talk on one, but draw on the other. All right, perfect. So with your protocells, basically adding clay parts is going to be common on early Earth, okay? So increases the rate of the vesicle formation. The vesicles will exhibit simple growth, mean they just get bigger. And they're able to reproduce by basically pinching off, sealing off, and making two vesicles. If you recall, think about what mitosis looks like. Okay, it's just a matter of pinching off and making two different cells. Well, the vesicles, they do the same thing. And so they can go ahead and they led up to vesicle reproduction and eventually led up to metabolism. That took a while. So they can absorb organic molecules, attach them to particles through selectively permeable membrane. And then eventually this led to the first self-replicating RNA. And so the first genetic material was thought to be RNA and not DNA. And so that RNA is going to play a central role in that protein synthesis. So it was a lot more simple because nowadays, that's funny, it is DNA. Let's see, trying to get my pen to attach and does not, oh, there we go. So now it is DNA is going to have the instructions to make RNA, and that RNA carries a complement of those instructions, and then that RNA is going to get translated into amino acids, and those amino acids eventually put them all together, they become proteins. Well, early on, it was a lot more simple. And so it was this system here. RNA started coding for early proteins. And so with that, in order to catalyze this, RNA molecules called ribozymes. So your ribozymes have been found now even to catalyze reactions. So it's thought that the ribozymes, which are small stretches of RNA, now they're larger stretches, but they were thought to start out as smaller stretches, that they were those original catalysts for reactions. Okay, so then your self-replicating RNA still. Your natural selection has produced self-replicating RNA. And through that, you get copying errors, just like with DNA. As you copy, you get errors. And so copying errors could have occurred, resulting in more RNA molecules present. And through that, that made RNA actually get, it says more adept, so more sophisticated, better at self-replicating because it started making more of it. And it was the beginning of the making of those molecules, starting out from small RNA pieces to becoming larger ones. And those RNA molecules that were more stable and replicated more quickly would have left the most decent RNA molecules. Those that didn't do well in their replication, they weren't stable, they're not the ones that continued on, because if they weren't stable, then they're not going to replicate and go to the next quote generation. So in 2013, researchers, they constructed a vesicle whose RNA would self-replicate within the vesicle. And so literally all you need, this is my take home for all of this, all you need are the building blocks. And what are the building blocks? Carbon, hydrogen, nitrogen, oxygen, sulfur, phosphorus. Those are the building blocks. And when I say all of those six, let's really say carbon, hydrogen, nitrogen, and oxygen. Those are the big guys, okay? So with that, if a vesicle on early Earth can grow and split and pass on RNA to its daughter cells, the daughters would be protocells. So if it can basically do that replication, these are the beginning of what we know of cells today. And so the most successful early protocells eventually, through natural selection, increased more and more and more, which will eventually lead to the early first cell. So let's go ahead and clear that. Your RNA could have provided a template for the formation of DNA, because RNA is single-stranded, okay? DNA is double-stranded. So double-stranded DNA is actually more chemically stable than RNA. So it is thought that eventually it led to the RNA becoming double-stranded itself, and then going to a more stable chemical molecule. Because as a reminder of what DNA and RNA are, so let's just go ahead and draw myself an RNA strand. So an RNA has a ribose sugar. I won't do a whole strand. This is a ribose sugar, okay? And a ribose sugar is going to be attached to a nitrogenous base. I'll just go ahead and do a double ring on that one, and a phosphate. Okay, so that is an RNA nucleotide. So we'll just say that this RNA nucleotide, let's go ahead and draw that RNA strand here. Very simple, adenine RNA, uracil, guanine, cytosine, and so forth. Put another C and so forth. So that's an RNA strand. So your DNA strands, it looks the exact same way, except for instead, let's go ahead and do a blue. If this were a DNA nucleotide, it would be deoxyribose. And the difference is literally the removal of an oxygen, okay? So your DNA strand would be a deoxyribose, but before we even got to that and had that removal of the oxygen, it is thought that two RNA strands, we went to what was called a double-stranded RNA strand. But because it wasn't as stable, it didn't last long. It wasn't ideal. RNA and an RNA. So this right here was thought to be the precursor to DNA, but it was thought that the stability of it wasn't good enough to continue on, and eventually led to the double-stranded DNA. So let's talk about the fossil record. The fossil record documents the history of life. And so basically you can see the changes being revealed all throughout the Earth's time. And so how does that happen? So your sedimentary rocks are going to be deposited into layers called strata. If you've ever, I've told you this on the last one, driven out to Woodland Park, and you drive on Highway 24 and you see those rocks on the walls, and they're actually like this. And you can see the obvious distinction of those rocks, right? And you can even see right here where it's even thicker because you have like an iron layer in there. Yeah. So those are called strata, is what we're looking at. And so sedimentary rocks, as your sediment, sounds just like it is. As your sediment is carried across a landscape and left there and deposited, it will harden and solidify and basically put on a new layer of rock. So you'll notice some of those layers are thick and some of those layers are thin, very thin. So it just really depends on how long and how much was laid down. So the fossil record is going to show changes in the kinds of organisms on Earth over time. And so few organisms have fossilized, and even fewer organisms did form that have been discovered. So what that means is what we have is barely even a drop in the bucket in what was really out there and over the course of time. And so that fossil record is going to be biased. And specifically, this is probably testable information you should know. It's going to favor species that existed over a long period of time, okay? That increases the chances of fossilization, or I should say of us finding a fossil of that organism, type of organism. Very abundant, lots of them, widespread. So not over the course of a long time, but over the course of a big geography. And then also the best fossils are going to be from organisms that had hard parts, such as shells and skeletons. Okay, soft body organisms do not fossilize well, or even at all. Some do, depending on the situation. But really, what you'll see, like we have some fossils of the homes of tube worms. Okay, so if you go out to not Woodland Park, excuse me, you think I'd love Woodland Park. If you go to Palmer Park and you drive up where you can do a little hiking in the around area, not to the top, but more in the bottom, and you'll see a little walk around area, and you will find these holes in the rock, and you will have lots of them, all of these holes in the rock, and they're hollow. You could literally take a stick and stick a stick in them. And what those are fossilized homes from tube worms. So the tube worms were killed, and the rock around them solidified. Well, the soft body of those tube worms eventually decomposed, but the hole in which they lived in, it's the remaining. So it's really cool. Go take a hike sometime, check it out. So the order of fossils in a rock strata, that's going to tell you the order in which they were formed. Okay, so they can figure out the ages of the fossils using this method, but you can't get the exact ages. It's actually going to be estimates. And so we use something called radiometric dating. And if you had Bio 111, you got to hear about radiometric dating and the half-life of carbon, because carbon 12 is very stable. Okay. You have carbon-13, which is an isotope, and you have carbon-14, which is also an isotope. So carbon-12 is very stable, but carbon-14 is not. It is not stable. So what it will do, it will decay. It will break down, and it has a specific half-life. And so if you look here at the chart of the half-lives, and so when you look at your radiometric dating, you can see this is the time in half-life. Time goes in this direction, the fraction of the parent isotope remaining. So what they'll do is they will check the amount of carbon-12 in a fossil and the amount of carbon-14 in a fossil. So because the amount of carbon-12 in the fossil is stable and it is expected not to decay, they don't really expect those numbers to change much. The carbon-14, on the other hand, they expect every 5,000 plus years it to half its life. So in about this amount of time, which is going to be this, I'm talking about carbon, okay? They also use radiometric dating with other atoms, because carbon is going to zero out. It's going to zero out a lot sooner than other isotopes and other atoms. And so this would be considered one half-life, where half of the carbon-14 is remaining. And then here's another half-life where it goes down by another half, and then it goes down by another half, until eventually it zeroes out and you end up with nothing left. And so when they take the ratio of how much carbon-14 is left in a fossil and they compare it to how much carbon-12 is left in a fossil, they can use math and they can go ahead and figure out how old that fossil is. This doesn't work for dinosaurs because that was more than 65 million years ago. And so your carbon will have half-lifed way before that. And so instead, what they do for other fossils is, oops, this is everything that I said. So your carbon dating will go up to 75,000 years. And then that's as old as you can use carbon dating. So instead, they will use other, like I said, other atoms, and then they will check the strata. You may have heard this before. So here's one strata, right? And say you have a bone. You may have already heard this, but I'm telling you again. And that bone is found in that middle strata. So they will check the half-life of molecules in this strata, okay? And then some have half-lives of hundreds of thousands of years of their atoms, excuse me. And they will check it in this strata. And then now they will have basically a little section. They can say it is between this many million years old and this many million years old. And that's why we can get a guesstimate. So with that, your geological record, it's going to be divided into different eons, okay? Hadean eon, Archean eon, and Proterozoic eon. Ooh, Phanerozoic eon also. So yeah, I'm not a connoisseur of learning all those words. I probably should be able to spit them out a lot quicker, but I can't. So just doing our best. All right, so with that, the Phanerozoic eon includes the last 500 million years, okay? And it is divided into 3 eras. And those 3 eras are going to be your Paleozoic, Mesozoic, and Cenozoic. And there's going to be major boundaries between those eras that correspond to big extinction events in the fossil record. Wow, look at that fossil record. Whew. So let's see if we can get in here a little. So let's start from the bottom. As you can see, we're going to start from here and go up. So with that, what are some of the numbers that I think are important to know? I think right here, the age of the Earth, approximately 6.4 billion years ago. Okay, I'm sorry, 4.6, excuse me, 4.6 billion years ago. That's an important number to know. Let's go ahead and talk about the other ones. You can see about 4 billion years ago, the oldest known rocks on Earth. And so basically solidification, where you got the rocks appearing on Earth. And then 3.5 billion years ago, the oldest fossils, so your prokaryotes. That right there, that's a good date right there. 3.5 billion years ago, with the oldest prokaryote fossils appeared. Then 2.7 billion years ago is going to be when you started to get Earth's, the oxygen in Earth's atmosphere began to increase. And the reason why these prokaryotes were responsible for increasing the amount of the oxygen in the Earth's atmosphere. So if you look at this though, between 3.5 billion years and 2.7 billion years, prokaryotes lived in the absence of large amounts of oxygen. They were anaerobic. And then some of those organisms actually produced oxygen, just like your plants would produce oxygen. And they're the ones who made our oxygen-rich atmosphere that we can live in today. So let's look at some other years. Oldest fossils of eukaryotic cells appear. That is 1.8 billion years ago. All righty. So that's an important, oh, it looks like I'm 1.8. That's what I'm trying to go for. My number's off a little bit. So just kind of going up. Diverse algae, soft-bodied invertebrates appeared. And so between the oldest eukaryotic fossils and your algae appearing, so your invertebrates are starting to diversify. They're starting to increase in amounts. They're still very simple, okay? Very soft-bodied and simple until the Cambrian period. So the Cambrian, if you've ever heard of this term, called the Cambrian explosion. The reason why it is called a Cambrian explosion is because there was a sudden increase in diversity among animal phyla. So this, they're not able to really determine the amount of species, but it's really the increased amount of phyla. So they call this the Cambrian explosion. And they had all kinds of crazy looking stuff, in my opinion. I think they're crazy looking. So if you ever seen Men in Black, these were real things. That was a real thing right there and right there. And so kind of cool. All righty. So then when we're still going up, remember going from the bottom to the top, your marine algae became abundant. Then you got the colonization of land by diverse fungi and plants and animals. So it is around this time period right here that organisms began to move from the water to the land. Okay. And then we start moving into vascular plants evolving, the evolution of vascular plants. So these are the dates here I'm most concerned with. You guys know, okay? At this point. Doesn't mean don't know the order of when vascularization came about in plants. You're definitely going to have to know that. But guess what? We have whole chapters on that. We got 2 big plant chapters. And so the next piece in the Devonian period, you're going to get a huge diversification of bony fish. Okay. And then the first tetrapods appeared. And those tetrapods then led to the next. Actually, let's go ahead and talk about the insects too. So this was a huge diversification event. Okay. Then if you go into the Carboniferous period, this was huge. The reason why you have gas in your car is because of the Carboniferous period. Okay. It is because there were huge amounts of forests and vascular plants on land. And because of the time on land at that time being swampy, plants falling over, falling in the swamps, and anaerobic environment, that caused your plants to decompose incompletely. And that incomplete decomposition led to your gas, your oil, your coal, all of that. So this Carboniferous period is very, very, very important. You're also going to see at this time you had that origin of your reptiles. Also, your amphibians became dominant at that time. And going up to the next, you will see the radiation of reptiles. Radiation, what does that mean? Diversification is the changing of the organisms. Radiation is them moving out into new areas and finding more niches and going far and finding other places to live. And so that's what that means. And so with that, you're also going to see extinctions take place. So as you notice, there's color change. And in these color changes, I'm going to go ahead and see if I can have a steady hand. This color change, I don't want to write over that. Boom. That was a huge change, solidification of the earth. And then you have this color change. Usually at every single color change, you end up having an extinction event, followed by a diversification and explosion event. That is something that you are going to see happening. Okay, because if you have a huge extinction event, guess what's going to happen? You got more land and more resources for other organisms to just take over. So let's go ahead and go to the next page. All righty, so we're going from the bottom, if you recall. Your next piece we're going to look at is about 252 million years ago, you started getting cone-bearing plants called gymnosperms, okay? And those gymnosperms, they're then going to give rise and lead to angiosperms, but angiosperms happen way up here, gymnosperms here. So angiosperms are flowering plants. Gymnosperms are all the plants that we just love in Colorado. Those ones, not the ones that planted in your front yard. You got those at Home Depot. I'm talking about the ones that are on the mountains, the ones that are naturally there. Those are mostly are your gymnosperms. Alrighty, so you can see here in your Triassic area that your dinosaurs have also evolved, right? And radiated, meaning they went out and found new areas. You had the origin of the mammals. But the thing about the mammals is they started out as tiny mammals, tiny nocturnal animals that lived underground, and they were not dominant at all, just originating. Okay, your dinosaurs are going to be ruling during this Triassic, Jurassic, Cretaceous. Okay, so with that, Jurassic specifically, that's why they named a whole movie out of it, Jurassic Park, those dinosaurs were at their most abundant and diverse. Okay, gymnosperms continued to dominate as the plants also. And in your Cretaceous, that's when your angiosperms came about, like I showed you. And your dinosaurs became extinct at the end of the Cretaceous. So what do we have here? We have this right here. It says 66. Just about every text I read say, and yours is going to say that too, that dinosaurs, they became extinct. Hang on. Sorry. 65 million years ago. So, trying to write here. So dinosaurs became extinct 65 million years ago. Boom. There you go. Hopefully you can see that. Because like it says, at the end of the period. Okay, so next, the Paleocene. The Paleocene, that's when you started to get mammals take over. Because like I told you, at the end of one extinction event, you end up with a diversification happening. So dinosaurs died and those mammals were like, can I come out and play? And they sure did. And they began to radiate. And from there, they began to make many different shapes, sizes, types, get into new niches, environments, all different kinds. You're also going to see the radiation of birds and pollinating insects. Okay. So, funny thing, I guess I should be specific, right? Because as I sit here and tell you that dinosaurs became extinct, right? Hang on, I'm trying to draw. Dinosaurs became extinct at the end at 65 million years ago. Well, not quite, because birds are the lineage of the dinosaurs. And so you're going to find when we get into that birds, we're going to have, when we get into reptiles, we're going to have reptile birds and non-reptile birds. And you're going to be like, what? Okay, so then let's see, what else do we have? In the Eocene, your angiosperms dominated. And then they also continued to just spread all over the place. Oligocene, you end up with the origin of the primates. In Miocene, you have continued radiation of your mammals and your angiosperms. So as you can see now, angiosperms are ruling everything. And they are. You just don't realize it. You don't realize what the angiosperms truly are. As much as I said on our mountain, it's all about the gymnosperms. We have a high amount of gymnosperms. But if you quit looking up and you look down, you're going to see the angiosperms are really running the show. Okay. So with that, let's go ahead and go to the next. Would I say the continued radiation of mammals and angiosperms? Look at this, 20, where are we? So we are looking at 23 million years ago, the earliest direct human ancestor, the earliest direct human ancestor. Okay, so when I say 23, it's really in this area between 5.3 to 23. And so 500,000 to 23 million. So that's a big little break there. Then you go into the appearance of your bipedal human ancestors. So this is going to be 200,000 to 500,000 in that little section there. And so then from there, origination of your genus Homo. So the genus Homo is, we are Homo sapiens. You have Homo erectus, Homo habilis, Homo habilis, excuse me, Homo neanderthalis, Homo nadali. So different homos, Homo is a genus, and sapiens is a species. You also have your ice ages right here. And then here we are in the now historical time. We wear jeans. So, all righty, let's see about next. I put okay, am I still recording? It appears like I am. Thought I put my moon on. All righty, so I got a phone call. It kind of kicked me off of what I was trying to do. Let's go ahead and get back on. Take a break if you need to. You could always just put pause. All righty, so let's move on. All right, so what we showed you in a nice little chart. You could also show it in an analogy of a clock. So the analogy of the clock is actually pretty cool when you think about all of from one start of the clock all the way to the finish, that'd be all of geological time on Earth. And so it looks like this. If you see right here being present time, see about getting us a little pin. So this right here being present time, okay, and this being the origin of the solar system and the Earth, okay? So your direction of your clock is going to go this way. Oops, let's go ahead and go for black on that one. It goes this way as you're looking at it. Do you see that? All righty. So everything that I just told you, you can put on here. So there's some significant things that you might notice. So some of those significant things you might notice would be, look at this. Prokaryotes have been around since the beginning, and they are still around. When you look at the present, they are still here. What else? Oxygen. As I was showing you, look at the time between your prokaryotes and oxygen becoming present in sufficient amounts on Earth. So those prokaryotes, they were anaerobic, and they are the ones that changed the Earth so that we could live. And so I'll just go ahead and let you look at this. Basically, we can go through and we can break it up, and we can go through all of it. I don't feel that it's necessary for me to go through all of it. I will hit some key points, okay? So with that, the oldest known fossils on Earth are called stromatolites. And those stromatolites are going to be rock formations from the accumulation of sedimentary layers on bacterial mats. These are so crazy cool. It is because of stromatolites, that is how we know that prokaryotes were the original soul inhabitants for more than 1.5 billion years on Earth. Because if you look right here, these are the stromatolites. These are the rocks, and they are essentially fossilized bacterial mats is what they are. Oh, we got somebody coming. Let's go ahead and stop this right here. Okay, I'm back. Sorry about that. So anyway, it is those stromatolites, all of those organisms, those are those bacteria that made life on Earth possible by putting out large amounts of oxygen. So with that, let's find you a perfect picture of it. So let's go ahead and share with you a cross section of those stromatolites. So this where we're looking in most of these places, this is Shark Bay in Western Australia. So if you look, let's find a cross section. I usually have a nice little, there's some fossil cross sections. What we're looking at here is the layers and layers and layers of bacterial mats that are laid down. So you can see them. So let's go ahead and put fossil stromatolites bacteria. just not given me. This is what I'm looking for. Perfect. So let's open image in new tab. So what we're looking at here is a cross section of those. And as you can see, here are the bacteria. So you can see all of the tiny little, let's go ahead and I'm going to do it in black so you can see the difference. So this is a bacteria and a bacteria and a bacteria and so forth. And it's going to have these huge chains of bacteria. Now, if you had 111, you might think that this looks a lot like cyanobacteria. And if you think that, then you would be thinking right, because this bacteria here is the ancestor to the cyanobacteria. And was responsible for putting out large amounts of oxygen into the atmosphere. Boom. All righty, let's go ahead and go into a new share, back to our PowerPoint. So I'll let you go ahead. Let's look at it. You can see where I showed you about oxygen becoming present, and it is a byproduct of photosynthesis. You will also see that that oxygen resulted in reactions with dissolved iron. So at that same time that happened, you're going to see a very large iron layer in the strata. If you recalled a few slides back when I drew the strata, I drew a really thick red one, and I told you that that was an iron layer. Well, that is an indication of the time period in which oxygen became very abundant on Earth. And so they call this the oxygen revolution, okay? And some groups survived in anaerobic environments, okay? But other bacteria, so there was so much bacteria present prior to this happening, most of them were anaerobic, but not all of them. So what happened was, when it got to a point where there was lots and lots of oxygen present in the atmosphere, basically bacteria had to do one or two things. They had to go find a different anaerobic environment, like guts of animals. You can find them in swamps. You can find them deep in the ocean. So they had to find anaerobic environments to live in. Or, I said one of two things. It was actually one of three things. Or they could adapt. If it was within their abilities to adapt, they could adapt to be able to use oxygen. Okay. And if neither one of those happened, they just died off. So there's actually a huge extinction event around that. I don't want to call it a whole event, but there was a large extinction that took place around a lot of those anaerobes not being able to survive in the new oxygen-rich environment. All righty. So this is just showing the oxygen-rich environment. So your first eukaryotes. Your oldest fossils of your eukaryotes is going to date back to 1.8 billion years ago. Your eukaryotic cells, they have a nuclear envelope. They have mitochondria, endoplasmic reticulum, and a cytoskeleton. That's what you can consider common for your eukaryotic cells. So it is thought, it's called the endosymbiotic theory, that your eukaryotes originated by this process called endosymbiosis. So this endosymbiosis is basically when a prokaryotic cell engulfed a smaller cell and then eventually evolved it into a mitochondria. So essentially, instead of phagocytizing it and eating it and digesting it, it phagocytized it and put it to work inside its body. And so an endosymbiont is a cell that lives within a host cell. So I have a picture of this on your D2L. I put it on your D2L because I guess I felt that the chapter didn't have it. They had it early on in your Bio 111 class, but they didn't have it so much here. So here is your endosymbiont theory right here. We will see it a little bit later, but I just wanted to refer you to this right here. Oh, let's go ahead and share. Didn't appear that I was sharing it. Let's try it again. There we go. Here's your endosymbiont theory. Go ahead and read that as you need to. Okay. So your single cell eukaryotes. Like I told you, anaerobic host cells would have benefited from endosymbionts that could use oxygen as it built up in the atmosphere. But over time, the host and the endosymbionts would have become interdependent, meaning they depended on each other. The host depended on the symbiote in order to provide it with resources, and then the symbiote depended on the host in order to provide it with a home and also access to resources. And so all of your eukaryotes are going to have mitochondria or remnants of mitochondria. Okay, so your plants, your animals, your protists, your fungi have mitochondria or at least some remnants of that. Okay, now another type of endosymbiont is a chloroplast. A chloroplast is not had by all your eukaryotes. Your chloroplasts are going to be exclusive to your plants and some protists. Not all protists, just some protists. So the term serial endosymbiosis supposes that mitochondria evolved before the plastids, like the plastids are going to be your chloroplasts and other energy related organelles. And so it suggests that mitochondria actually evolved first. And so mitochondria in your plastids likely descended from those bacterial cells that got phagocytized, and the original host is thought to be archaean or a close relative. So what I'm trying to tell you is archaea is thought to be the original host and not so much the bacteria. Oh, here we go. Here's the beautiful picture that I was looking for. So this is just going to show you your ancestral prokaryote and how the basically the building of the membranes, the internal membranes. And so when you look here, you can see your DNA inside, just like an ancestral prokaryote. Okay, you have your plasma membrane, just like prokaryotes have a plasma membrane. But through the inner folds of the plasma membrane, it's thought that these inner folds eventually touched each other and became the nucleus, the nuclear membrane. And so you can see more inner folds making here, which led to become the endoplasmic reticulum, the rough endoplasmic and the smooth endoplasmic reticulum. And then eventually you can see the engulfed aerobic bacterium. Uh-oh, it won't go away. This has happened before. I don't know why. Sometimes it does this. What I have found is I have to stop sharing in order to make that happen. Let's see if I can share something else instead, make it change. Oh, looks like that worked. Perfect. All righty, so then same thing. Mitochondria being engulfed, that led to the ancestral heterotrophic organism, one that needed to acquire energy from another organism. And then with plastids and chloroplasts being made, engulfed, that led to the early photosynthetic eukaryotic organism, which, for example, is going to be your plants or your protists. So your first eukaryotes, there's key evidence supporting that endosymbiotic origin of your mitochondria and your plastids. And so first, like I told you, your inner membranes of both of your organelles are going to be similar to plasma membranes of living bacteria. And I said, like I told you, I'm so sorry. Like I told my Bio 111 class, that the membranes of the inner membranes of the bacteria are similar to that of the mitochondria and chloroplast. You're also going to find DNA structure and cell division are similar to bacteria. So your mitochondria and your chloroplast have DNA, and they also undergo cell division really almost identical to that of bacteria. And then so both organelles are going to transcribe and translate their own DNA. So your mitochondria and your chloroplasts, they will divide independently of the host cell, and they will transcribe and translate their own DNA, which leads to the next thing, they make their own proteins. So this right here is continued evidence for the endosymbiotic theory. So with this, if I were you, I would put a big star on this slide. This is really going to sum up everything. So that evolution of your eukaryotic cells, that allows for a greater range of unicellular forms. Okay, so the fact that unicellular cells evolved, that basically opened up new opportunity, lots of opportunity for new unicellular forms. And so that led to what is a second wave of diversification. And that is the evolution of multicellularity, which eventually gave rise to algae, plants, fungi, and animals. Sorry, just moving rooms. Okay. So when it comes to your early multicellular eukaryotes, big take homes on this, they started to evolve. The oldest fossils found are small red algae, about 1.2 billion years ago. Older fossils from about 1.8 billion years ago may also be multicellular eukaryotes, but they just haven't, like it says, resolved them taxonomically. So they stick with the best evidence that they have right now. Leading to that Cambrian explosion. Okay, so I just showed you the period of time, but I did not give you the years. So now I'm going to give you those years. Those years that are important to know is 535 to 525 million years ago. So the Cambrian explosion took place over the course of 10 million years. And that is that sudden appearance. Yeah, that's sudden, 10 million years. Well, considering how long it takes to evolve and the amount of evolution that actually took place, yes, that is considered sudden. And so there's lots of different, more modern animal phyla, and a few phyla appeared even earlier, like your sponges, cnidarians, and mollusks. So look at that. We're getting really close to the present time here. So as you look here, this is a nice little clade that takes you to the diversification of what leads to animals, okay? So animal diversification. If you see, you can see take it off a highlighter, put it on a pen, there you go. This is going to be the common ancestor right around here. That's where the split of lineage went. The common ancestor went to sponges, okay? And the reason why I'm calling this a clade is because, yes, this is a phylogenetic tree, but this specifically, I could call this a clade because the sponges I can consider an outgroup. What you're gonna learn in later chapters is that sponges do not have true tissues, and everything else does. So these are the animals, and the sponges do not have true tissues. And so they look and are the same as the early representatives that evolved 695 million years ago. So that's pretty awesome. That speaks a lot. And so you can see next common ancestor led to that diversification of your cnidarians. And then that lineage split, which eventually led to your echinoderms direction, and then also the direction, so your echinoderms and your chordates, and then the other direction led to your arthropods, mollusks, your annelids, brachiopods. So here's the interesting thing. You would maybe not even think about this. We're chordates. We are closer in lineage to a sea star, not a starfish, a sea star. We are closer in lineage to a sea star than we are a lobster. And you might have thought because the lobster had arms and legs that it had opposed like appendages, you would think that we'd be closer to that. But nope, we're closer to a sea star. All righty, some good Trivial Pursuit for your Friday night. So that Cambrian explosion, that provides that first evidence of predator-prey interactions. And so this is basically where there was lots going on. There were lots of predator body shapes. There were lots of prey body shapes. Prey could be predators to other things and vice versa. And everything was just pretty much eating everything else. Okay. Large body predators with claws and other adaptations for catching prey, they started to appear in that fossil record during that time. You also started to see lots of defense adaptations. You started to see sharp spines, body armor, all kinds of crazy stuff. And through DNA analysis, that's going to suggest, like I told you before, that your sponges evolved 700 million years ago. And that common ancestor to several other phyla lived 670 million years ago. And so your sponge is going to be, more of the oldest and less changed, I should say that, the less changed type of organism all throughout time. The oldest fossil assigned to the extant animal phyla lived 560 million years ago. We say extant, not extinct, meaning we have representatives of them around now. We have found fossils and represented now. So extant, not extinct. Just drinking some water so I don't cough at you. Alrighty, so the colonization of land. Fungi, plants, and animals started to colonize land about 500 million years ago. Okay, so many of your plants evolved adaptations. This is huge. We're going to have whole chapters on plants evolving adaptations to be able to come to land. But the big take home is going to be vascularity. The fact that plants could evolve a vascular system. This helped them keep from drying out. And so they did not need to be so dependent upon the water. They can find themselves further away from the water. Okay. They still need water, but you're going to find vascularity is huge in evolving the ability for plants to, if you think about our plants, they're nowhere near a large body of water. The only water they get is when we turn on our sprinklers. So it is thought that your plants and fungi likely colonized land together. And fossilized plants will show evidence of mutually beneficial associations. We're going to hear this in more than one chapter. Okay. They are called mycorrhizae. It is thought that your fungi evolved into a symbiotic relationship with plants, not all fungi, but a specific fungi called mycorrhizae. And that symbiotic relationship helps the plant obtain water. So if you were to pull a plant out the ground and look at its roots, you might see little fungal bodies around it, little white fungal bodies. Those are called mycorrhizae, and they help the plant obtain certain nutrients from the soil and water from the soil. And in turn, the plant will feed the mycorrhizae sugar. And so they have what is called a symbiotic relationship. And we still see that today. So through that colonization of land, that's leading to arthropods, tetrapods. They're going to be the most widespread and diverse land animals. Your tetrapods, they evolved from lobe-finned fishes. And that evolution took place around 365 million years ago. Okay. That lobe-finned fish made it possible for those fish that maybe got washed up into shore, they got left in tidal pools, and then needed to go from tidal pool to tidal pool. So there's two things going on here. Those fish, not only lobe-finned fish, were able to get from tidal pool to tidal pool. How were they able to get there? They actually had stronger bony fins that helped to move them on land to get into another tidal pool. And because they were lobe fin, this is the beginning of the quote unquote lung. And so the human lineage of tetrapods that evolved about 6 to 7 million years ago. Modern humans originated 195,000 years ago. Okay, so the genus Homo. So your history of life on Earth, it has seen that rise and that fall of many different organisms. So what that means is the organisms we see today, not many of them were around from the beginning. The only ones that we can really say are a huge group of prokaryotes that were around from not even the beginning, but I should say the first evidence of life. And so those rise and those falls, those groups, lots of different groups arose throughout time and then went extinct. And so that depended on speciation and extinction rates within your group. And so how did extinction rates affect diversity? And so this is just a generalized chart showing a common ancestor and how you can have lineage A, and it could go in a direction of lineage B. And so as you can see here, lineage A fall out. They were unsuccessful during their time. And so this is at 0 million years ago. Well, this one was successful. Let's fix that one. But all of the other ones didn't make it to time 0, which is current time. Okay, so when you look at lineage B, these ones, this was successful. Get my pen going. Successful to current time. They made it. They fell out. They made it. Yep. And so forth. Boom. Okay. So this is a perfect stopping point. I will go ahead and pick up with part 2 of your video.